Hi, everyone, and welcome to today's What's Happening in PWS Spring webinar. I'm Melissa Demand, and I'm the Director of Community Engagement for the Foundation for Prader-Willi Research, and I will be today's moderator. Today, Dr. Teresa Strong will give you updates on some of FPWR's current initiatives, as well as details on the 9 grants that were recently selected for funding. The work we're going to share today has been made possible by the generous support of our community. So I want to take this time to thank our donors and fundraisers for your incredible dedication to helping fund PWS research. We have a lot of information to share with you today, but please know that we will save time at the end of Teresa's presentation for questions. So feel free to drop any questions you may have into the question panel, and we'll try to answer as many as possible following the presentation. And with that, I'm going to hand things over to our Director of Research Programs, Dr. Teresa Strong.
Great. Thank you so much, Melissa. Welcome, everybody. It's great to see everyone today. I am excited to share some of the progress that has gone on over the last couple of months in PWS research. And as Melissa said, tell you a little bit about the new grants that we're funding. So these are grants that were submitted last fall and were selected for funding at the beginning of the year. So we'll do an update on just sort of the general research activities that are going on in the community, then review those awards. And then, as Melissa said, if you have any questions on any topic related to PWS research, please feel free to drop those into the Q&A and we'll get to those at the end. Just as a reminder, our mission is to eliminate the challenges of Prader-Willi syndrome and Chaff-Yang syndrome through the advancement of research and therapeutic development. And this is a diagram I use quite a bit to show the progress from research and discovery all the way through to the development of new therapies, the testing of new therapies, and then what we call post-approval, which is how to apply those therapies most effectively. And the grant program that FPWR has really it supports research across this entire spectrum. So everything from discovery, where we're really trying to understand what are the differences between PWS and, for example, typical neurons, all the way through trying out different compounds that may have beneficial effects in PWS, in model systems, and then in humans. So our goal is to accelerate every step of this process by building resources that all the research community members can use, fostering collaborations, supporting innovation that allows these things to progress more quickly. So I'd be remiss if I didn't start off by talking about the biggest news over the past couple of months, which is the approval of the first drug, the first FDA approval for a drug to treat hyperphagia in Prader-Willi syndrome. So VYKAT XR has been approved for individuals with PWS age 4 and up to treat hyperphagia. And you can see there is a website. I'm sure most of you have heard about this already. It's Soleno Therapeutics drug, and there's information on that website about prescribing information and resources on how to access the drug. But it really is a testament to the entire PWS community that this drug has been approved. Of course, we appreciate Soleno and their hard work and their persistence in seeing this drug through to approval. But we also could not have done it without the families who were involved in the clinical trial, without the dedication of all the clinicians and their the clinical site people who made that trial go even through COVID, as well as all of us who maybe participated in the global registry in the Path for PWS Natural History Study. That data was critical as well, helping to support and show the efficacy of VYKAT. Anyone in the community, who were many of us, who told our story to the FDA and spoke about the impact of PWS so that the regulators at the FDA could understand, the potential benefits of this treatment. So it really is, it was a community-wide effort, and it's really great to see that reach this point. But I do want to highlight that the particular roles that FPWR had in that process. So FPWR did fund the initial study of DCCR, which is now called VICAT, in individuals with PWS way back in 2014 before Soleno even existed. And as I mentioned, your participation in the Global PWS Registry provided a background information about the natural history of PWS that was really critical for many aspects of that study, and the path for PWS study in particular. The data from that provided supportive data to show the efficacy of VICAT. And then all of the advocating and again, sharing with the FDA in partnership with FPWR, in partnership with PWSA, and all of the other advocacy efforts, individual groups in the PWS community to get the points across about the unmet need in PWS. So this is fantastic, but our work is not done yet. So I have 3 slides of how our work continues. So as VICAT has been approved, we've seen there's been a lot of chatter, of course. Does it work for everybody? Who benefits the most? How long does it take to see an effect? What kind of changes can we expect to see in our, loved one with PWS? How long does the insurance take to approve it? And I think we as a community, particularly those of us in the US who now have access to this drug potentially, really have a responsibility to others, to those who come after us, to really document and understand the answers to these questions. So one way of doing that is by, we're going to put a new module into the Global PWS Registry, which will allow individuals who have been prescribed VICAT to answer these questions. So as we know, the clinical trial was a very controlled situation. It was less than 100 individuals. Now it's going out into a much larger population. So we really want to capture the experience in the real world and have that information to see how we can best optimize application of VICAT XR. There'll also be an effort with the PWS CLIC, that's the Clinical Investigative Collaborative, a network of PWS experts in North America. And they will also be gathering data on the use of VICAT and seeing how best, again, to use that medication to the optimal benefit of everyone with PWS. So please look for those efforts. Please participate in those efforts. It's only by your participation that we can really understand how best to use this medication in PWS. Our second work continues slide is about additional therapies for PWS. So VICAT XR is fabulous. We're really thrilled that it is now on the market. However, we know that it's not going to be the perfect drug for every individual with PWS, and we want lots of different tools in the toolbox. So there are ongoing PWS clinical trials that may be appropriate for your loved one with PWS. For example, if your person with PWS struggles with temper outbursts, you may be interested in trying the VNS for PWS clinical trial, which looks at a device, a vagus nerve stimulator. Acadia Pharmaceuticals, their ongoing study for looking at carbetocin in PWS, I believe, is nearing its enrollment goal. So we're very excited to see how that moves ahead and looking for those results, I believe, in 2026. Harmony Biosciences has an ongoing trial with pitolisant, which is a drug that is meant to improve daytime sleepiness in PWS. So if you have a child with PWS who is struggling in school because they can't stay awake and learn, you might want to learn more about that clinical trial. And Aardvark Therapeutics has a novel drug that works in the gut and reduces appetite, or we think it may reduce appetite. The clinical trial is to demonstrate whether it is efficacious, whether there's efficacy in that. So Those are just some of the clinical trials that are ongoing. There's also several small trials ongoing at different academic centers. Please visit the PWS Clinical Trials site to learn more about that and sign up for the Clinical Trials Alert. And really, it would be great to see some of these other therapeutics move forward quickly as well. The final aspect of our work continues is other ways to support research. So here are three efforts that are sort of one-time, relatively brief commitments that will support different aspects of research. So the Vanderbilt group is developing a self-report, so individuals with PWS themselves reporting on their hyperphagia. And they need families, particularly the individual with PWS, age 10 and up, to go through this questionnaire and provide feedback. So it's a great thing. It sort of empowers the person with PWS to talk about their experience with hyperphagia. It's brief. It's a 15-minute Zoom call. Elizabeth's team is fabulous. So I would encourage you, if you have a child or young adult with PWS, regardless of whether they have severe hyperphagia or mild hyperphagia, because really the Vanderbilt team wants to see how this works across all individuals with PWS, to set that up. Dr. Dimitropoulos has been looking at play-based interventions in children with PWS to see if that improves their social abilities later in life. And she's doing a follow-up study. Whether or not you have been through that program, she would like individuals age 8 to 20 to do a quick, again, a relatively brief one-time Zoom call to get some feedback on some of the aspects of that program. And another effort is looking at educational trends and support. So your experience with the education system. Dr. Pacha at the Chicago School is running that. That's a survey-based investigation. So if everybody on this call were to do one of these studies, whichever one, if you have a child age 3 all the way through an adult age 60 with PWS, if you choose one of these and do it, all of these investigators could get their study done really, really quickly. So we'd love to see them be able to do that. Okay, so with that, I will move to the FPWR grant program. And this has been kind of the bread and butter of the organization since we first started. And over the past 20 or so years, we've invested, thanks to all of you who have done the fundraising and made the donations, we've invested more than $20 million in research, as I said, across the spectrum. And that has resulted in some new insight into the function of the genes in the PWS region, an improved understanding of our appetite in PWS, new cell and animal models that lets researchers study PWS more effectively, new targets, clinical trials, and optimization of clinical care. And really importantly, it has supported new investigators coming into the PWS space who are interested in working and learning about PWS. So there have been many accomplishments. I just want to mention a few of the outcomes that have come out just in the last couple of months. And you can see some of the publications here, and I'll just walk through them pretty quickly. So two of them that I'd like to start with are different measures that are relevant for families with PWS. And those measures can be used in clinical trials as well as to understand how effective treatments are. So those are the validation of the Food Safe Zone questionnaire by the group at Vanderbilt. So this is a questionnaire that measures food security at home. So it measures what we do. Do we lock the cabinets? Do we monitor? How well do we supervise our kids with PWS, kids and young adults with PWS? So it's a questionnaire, and it gets scored. And this paper really describes the development of that questionnaire and the analysis of sort of how well it works in the population. And what Dr. Dykens and our colleagues found was that the food safe zone scores, or the kind of the work we do to keep our kids safe from food, increases with age for individuals with PWS across all of the domains. So supervising, avoiding food situations, locking food up, for example. And the food safe zone measure adds context to the other assessments. So when we're talking about hyperphagia behaviors, or the things that we see because individuals with PWS have a high degree of drive to food seek, it helps to understand what kind of environment they're in day to day. As those behaviors show up. It also is a good for letting pharmaceutical companies or the FDA understand what we do as families to try to keep our kids safe from food situations. The other questionnaire, and both of these really got just a ton of data through the Global PWS Registry. So if you fill these out through the Global PWS Registry, thank you so much. We really could not have done it without your participation. So from the FPWR team, led by Lauren Schwartz, we published a study on life satisfaction, global health, and mood in PWS. And this study used some well-used, sort of validated measures that have been used in the typical population. They've been used in other intellectual disabilities to measure mood, life satisfaction, global health. And we wanted to do this so that we had a nice baseline of where individuals with PWS are on those measures, so that as we get new treatments on board, we can re-ask those questions and see if the quality of life is improving. So validation of these measures in our population just gives us some resources that are widely known in the research community. Another study from the group in France, so Drs. Barrett and Muscatelli developed a new mouse model of PWS, and this mouse model has two important genes, MAGEL2 and nectin, both deleted. So there's been a nectin deleted mouse, there's been a MAGEL2 deleted mouse, and now both are missing. And that's important because these genes kind of are, they work together. So now we've got one that is closer to the PWS phenotype. And they characterized this mouse model and showed that it has many features consistent with PWS. So they have that failure to thrive early on in the shortly after they're born. They have respiratory abnormalities, so they have a lot of apnea. They have a disruption in their circadian rhythm. They have impaired maturation of their oxytocin neurons, and then social changes probably because of that. So this mouse model is a new mouse model that the research community will find very useful for investigating things like the role of oxytocin in the development of the brain, and what therapies one can use to overcome some of the social deficits. Another more clinical paper is from Dr. Lucarelli, who is at Mass General, and he has been looking at the hospital records. So there's a massive database of all the hospital records in the US that allows you to look at individuals who are diagnosed with PWS but have other things going on. And in this case, he looked back over the COVID years, the time when the pandemic was the worst, and asked, how did people with PWS fare compared to other populations? And what he found that was individuals with PWS who were hospitalized with COVID had a much harder time. So they had to stay in the hospital longer. They were more likely to have to go on a ventilator, and they were more likely to pass away. So this is interesting because anecdotally, sometimes we hear, oh, everyone in the family got COVID. My son with PWS got it, but he didn't seem to be as affected as everyone else. And that may be true, but I think overall the point is I've noticed with my young adults with PWS, you may have noticed with yours, sometimes it's hard to tell when they're really ill. And I think we have to be very careful, especially about respiratory disorders in PWS. So this paper suggests that it's important to continue to take precautionary measures and stay up to date on vaccines. Another more lab-based study looked at circadian rhythm. We've talked about circadian rhythm, which is disrupted in PWS, and it's disrupted in people with PWS, it's disrupted in mice with PWS, and if you grow cells in a lab dish from a person with PWS, you can actually measure that disruption in the circadian rhythm. And that's what Dr. Reiter and his team did. They showed that there's disruptions in cells growing in a dish. And this is important. And importantly, they showed that they can use drugs to correct that disruption in the circadian rhythm. So that's important. The drugs that they used aren't good, to use in the clinic, but it gives them a starting point at which to find drugs that might be good clinically, that can be used to reset this, potentially to reset circadian rhythm. And that's important because not only is it, things like waking up at night and falling asleep during the day, that's kind of disruptive for social reasons. But when your circadian rhythm is disrupted like that, it really impacts negatively things like metabolism and cognition. So it would be really good to be able to get that circadian rhythm back in line in individuals with PWS. The final recent outcome that I'll discuss is a study from the Gershbach lab at Duke University, and they are experts in the use of CRISPR technology. So this is one of those great examples where funding from FPWR allowed them to take their expertise, which is CRISPR, and really apply it to the problem of how would you use CRISPR technology to do genetic therapy in PWS. So they were looking at lots of different approaches to do CRISPR, because there's many different ways you can use that technology. And they were trying to identify what is the best at doing gene activation, so turning on the maternal chromosome, which is present in every individual with PWS, whether you have deletion or imprinting mutation or UPD. How do you turn that maternal chromosome on? And importantly, how do you keep it on? Because it's not going to be only turning it on once. It's going to have to stay on for the life of the individual, presumably, anyway. So they were able to achieve long-term stable expression of the PWS genes in stem cells and differentiated cells that had been encouraged to make neurons in a dish. And this is an important proof of concept and a first step. We have funded them again now to look at ways of activating that gene expression in fully differentiated neurons, so at a later stage of maturation, which will be important to go into the clinic. But this was a great proof of concept and first step for CRISPR gene therapy for PWS. Okay, so that is all of the recent outcomes from previously funded grants. Now I'm going to turn to talking about the 9 new grants that have been funded. And again, these grants kind of cross the spectrum all the way from very basic science to, understanding things more in the clinical sense. So in total, these grants are about $1.2 million. So there are 4 grants in the category of genetics and cell biology, 3 developing new targets, potential targets for PWS, and then 2 in the area of moving those targets forward or applying them to clinical situations. So first I'll talk about the very basic science one, and I'll stay high level, I hope, on this. We have 2 projects, in fact, that are looking at the SNORD genes. These are the genes that we know are very important in PWS. Every person with PWS is missing these genes. We don't know exactly what they do, and they've been very difficult to study because they're not kind of the genes that you learn about in your biology class. They're not genes that make proteins so that we can study the proteins. They're genes that make funky RNAs that then go off and do things that we don't quite understand. So Dr. He at the University of Chicago has made it his life's work to understand the function of these unusual RNAs, and he's applying his expertise to PWS now. So whereas there has been a lack of understanding about what are the SNORD115 and SNORD116 genes, which are in the PWS region, what what they're interacting with in the cell. He has some novel technology to map the targets of those genes and understand, importantly, how when we lose those genes, as in PWS, the cell changes. So we're excited about this research. This is actually a renewal. We funded him before. He's been making excellent progress. He's an outstanding researcher. He's a leader in the field of RNA biology. And the work that he is doing has the potential to significantly enhance our understanding of the role of these SNO RNAs, which are encoded by the SNORD in the PWS locus. So the long-term goal of the study is to advance our understanding of the normal function of SNORD genes and establish how cells change when those SNO RNAs aren't present anymore, as is the case in PWS. And this knowledge is really important because it can help guide the development of new therapies that might compensate for those missing SNO RNAs. A second study from Dr. Amanda Whipple at Harvard University is also looking at the SNORD116 RNAs. She initially looked at them in mouse cells and found some of their binding partners and started to get a sense of what they were doing. And with funding from FPWR, she is now looking at them in human cells, looking at the human SNORD116 in human cells to see if it matches what she's been seeing in the mouse cells. And she is another person who spends her life looking at RNAs, studying RNAs, which I have to admire because I think it's not an easy thing to do. And they have some methods that they've developed as well to look at what these RNAs are doing. So she's applying those optimized methods to human neurons, and asking whether that interaction of SNORD116, particularly with ribosomes, and ribosomes are the machinery that makes proteins within the cells, is the same in human as she's seen in mouse. The impact, again, of losing SNORD116, which happens in PWS, will be assessed, and its impact on protein production in particular. So we're excited about this. This is an enthusiastic researcher with RNA and neurobiology expertise. She's using innovative and highly sensitive cutting-edge approaches to address this question, and she has promising results in her initial studies. The long-term goal is to characterize the normal molecular function of SNORD116 and the consequences of its loss. And that, again, will allow us to be smarter about developing therapies to compensate for the loss of those genes in PWS. The third grant that we have to discuss today is looking at the molecular mechanisms of neuronal development and the deficits that occur in PWS. And this is using insights, this is using organoids to look at hypothalamic function. So organoids are sometimes called brain in a dish, right? So you can grow cells from an individual with PWS, and in this case, they've actually been engineered. This group has been working on PWS for several years now, and they've engineered some neurons that essentially have PWS. You can grow them out in 2 dimensions, but organoids are when you grow them into 3 dimensions and allow them to form a little brain. In this case, you can add growth factors, so they're essentially forming a hypothalamus. And then you can study how the cells in the hypothalamus in a dish are interacting. So the goal of the study is to understand the different cells in that organoid and how they're interacting as a hypothalamus in PWS. We're excited about this. As I mentioned, this group has been looking at PWS for several years now. They've developed a number of cell lines that they can reliably differentiate into neurons in a dish. They have a ton of resources with respect to the most advanced technology to look at the RNA expressed by all of these cells. The organoids are really an interesting model, not only to understand how cells interact, but also there's been a lot of progress in looking at drug development using organoids. So this is setting us up potentially to develop or test drugs. Essentially, you'd be testing it in a little brain in a dish. So the long-term goal of this study, so the data from this study should provide insight into how the hypothalamus in PWS differs from the hypothalamus in typical individuals. And the data will serve as a baseline for later drug testing in this organoid model. Okay, the next study is a very basic science study from the Alzheimer lab at Heidelberg University. So they are using a rat model of Schaff-Yang syndrome. It's also a reasonable model for Prader-Willi syndrome. So it has a disruption in the MAGEL2 gene. And they're going to be studying some really basic questions that have not been fully answered yet. And this actually comes out of the MAGEL2 roundtable. So all of the researchers who study MAGEL2 get together and they talk about kind of what are the missing pieces in their field, like the basic pieces that everybody could use to knowledge on. And one of those questions was, we don't know exactly how MAGEL2 is expressed in the brain throughout development. So Dr. Alzheimer has taken on that challenge, and as well as understanding during circadian rhythm how MAGEL2 expression changes. They'll be measuring MAGEL2 gene expression in the brain of this rat model from embryo all the way through adulthood, and also with respect to circadian cycle. So we're excited about this because this is missing information that everybody in the field agrees needs to be understood. And it has the potential not only just to better understand how this gene works, but also to think about, okay, do we need to be concerned about the timing of a therapy for Schaff-Yang syndrome or Prader-Willi syndrome? Is there a time where it has to happen in order to have its effect? The long-term goal of this study is to guide the development of therapies and help determine if there is that optimal timing for treatments to overcome MAGEL2 mutations or deficiencies. Okay, we're moving now onto some more preclinical studies looking at potential disruptions in a whole either animal or human rather than at the cellular level. So the first grant in this area is neurovascular plasticity as a molecular driver of PWS. So neuroplasticity in science just means, it's flexibility essentially. So the neurovasculature, or the blood vessels that feed into the brain, can be really important in regulating things like neuroinflammation, cognition, and metabolism. The brain is an organ, and it needs energy as well, and it needs a good solid vasculature for that. So Dr. Scheenberger Payne is an expert in this neurovasculature and how it impacts a whole being. And it really has not been looked at in PWS, and there's reason to think that it might be different in PWS than in typical individuals. So the goal of this study is to describe the vascular topology, the blood flow dynamics, and the blood-brain barrier permeability in a mouse model of PWS. We're excited about this project because this lab is a leading lab in looking at imaging tools to understand the neurovasculature. And this is an interesting, it's not a very well studied area of investigation that could have relevance to PWS. The long-term goal of this study is to look at drugs that might correct neovascular dysfunction if it's found in PWS, and that might have the ability to correct multiple clinical symptoms at once. Another area of preclinical development is from Dr. Moynes. lab, and they have looked at this protein DGKK. And DGKK has been shown now by multiple labs to potentially be a downstream target of SNORD116. So when we lose SNORD116, we get alterations in DGKK. And that's important because DGKK is a protein that is important in metabolism and regulating body weight, among other things. So this study will determine if and how DGKK is affected in mice that are missing SNORD116. And it'll look at their blood and brain and metabolism. And it will also look at human neurons in a dish to see if the same is occurring in humans and in mice. So we're excited about this study because it is a compelling potential downstream target of SNORD116. So it may be one of those things that SNORD116 regulates. And it's linked to multiple aspects that would be important in PWS, like metabolism and behavior. There are drugs that exist to modulate DGKK. So if this is found to be a target, we have the potential pathway to a clinical product that may be able to influence that. And the long-term goal is to overcome SNORD116 deletion in PWS by using drugs that can compensate for that SNORD116 loss by targeting DGKK. Another study, this is also a renewal from Dr. Vetravelan, is looking at sleep in PWS. We know, as we've said multiple times now, that sleep is disrupted in PWS, and that can have long-ranging consequences, not only for the parents who have to wake up in the middle of the night, but also for metabolism, cognition, and behavior in PWS. So we know that people with PWS, have daytime sleepiness. They have kind of this just disrupted sleep pattern. They may not really differ in the number of hours that they sleep, but they have the sleep-wake instability. So they wake up more at night, they sleep more during the day. And these MCH neurons, the melanin concentrating neurons, may be overactive, and that may be part of the problem that's driving this in sleep-wake instability. This study will use neural activities activators of MCH to measure the activity across sleep-wake cycles in a mouse model of PWS and examine the alterations of the MCH system in PWS. We're excited about this study because this is an experienced sleep researcher, so he really knows these systems and measures well. Sleep issues in PWS are an unmet medical need that are likely, as we've said, to impact behavior, mental health, metabolism, many aspects of development. The long-term goal of this study is to examine dysfunction of the MCH system in PWS to help identify new drugs that can manage sleep and circadian rhythms in PWS. And finally, now moving into the things that are a little bit more clinically relevant from Dr. David Godler, who's been working in PWS for several years now. He's been looking specifically at whether we can find blood markers that will predict the severity of PWS. So, we can't go in and sample someone's brain, but can we find some blood markers that will allow us to identify who might be at highest risk, for example, for psychiatric disorders, so that we can identify those risks early and go on a path that will mitigate those risks. So the study rationale for this study is that biological predictors for symptom severity really haven't been defined yet. And Dr. Godler has been looking at this question. He's identified at least one gene that looks like it may be, a potential predictor in the blood of what's going on in the brain. So the goal is to analyze PWS, now look in brain samples from individuals who have passed away to understand how the abnormal activity of these genes that he's identified influences the expression of other genes in different cell types that may be contributing to the PWS symptoms and severity. We're excited about this study because this is an experienced PWS researcher. They have strong preliminary data linking the blood and brain marker. They're using some cutting-edge technology and really having that information about who is at risk for what which of the symptoms of PWS would be really helpful for families, again, to try to get ahead of some of the potential concerns. So the long-term goal of this study is to define the genetic pathways that are dysregulated in PWS blood and brain cells that are clinically significant and may inform the care of individuals with PWS. The other thing is, by identifying these pathways, Dr. Godler is also interested in looking at what are the drugs that already exist that may be able to influence these pathways. And for example, if you can identify those who are at highest risk, is there a drug that you can then use to reduce that risk? Okay, the final grant that I'd like to mention is from Dr. Yi at Amsterdam University Medical Center, and they are looking at mechanisms of cholinergic neuronal impairment in individuals with PWS. So they have looked at brain samples of individuals with PWS, and they have found that a specific neuron that makes these cholinergic neurons look to be deficient in PWS. So the cholinergic system of the brain is involved in lots of things. So learning, memory, attention, mood, sleep, and appetite, obviously all of which are relevant to PWS. And what she's found to date is that PWS brains have fewer acetylcholine synthesizing neurons. So the goal of this study is to investigate that further and correlate the levels of acetylcholine in individuals with PWS, living individuals with PWS, with cognitive and behavioral assessments. And that will establish whether it makes sense to use acetylcholine enhancing drugs, which are available, as a potential treatment for PWS. So we're excited about this project because it's an exceptional team of investigators that have experience both in basic and clinical science and in specifically in PWS. They're using some cutting-edge brain imaging technology to look at the cholinergic system in PWS. This system is potentially targeted to multiple PWS symptoms, as we've talked about, like behavior, appetite, cognition. And we have the potential here to repurpose already approved drugs if this pathway is demonstrated to be really relevant in PWS. So the long-term goal is to determine whether treating PWS-associated acetylcholine deficiency improves cognitive deficits and appetite-related changes in PWS. So this is a very exciting project that is getting underway. Actually, they're all very exciting projects. We're really thrilled with this round of grants. It's just a fantastic group of investigators. I think, we've really been fortunate to be able to attract such great investigators to be working on PWS. Okay, so I think we'll wrap up here and take your questions. Looking ahead this year, very exciting year. Again, we'll be expanding the global registry, PWS registry, to include questions about VYCAT. I would encourage you all, again, to please participate in that so that we can learn as quickly as possible how best to use that medication. We'll be getting a facelift on the global registry this year, so that's exciting as well. We've launched the Shaft-Yang syndrome registry. That's very exciting for those individuals in our community who have Shaft-Yang syndrome. Please, again, remember that there are many ongoing clinical trials, and we'd love to get as many tools in the toolbox as we can. So we know that PWS is very diverse. There's all kinds of different symptoms that need to be addressed. And the more medications, devices, behavioral interventions that we have at our disposal, the better the life for the individual with PWS is going to be. We're also looking this year at the PWS CLIC, the Clinical Investigation Network. They are building their clinical database, which complements the global PWS registry. So in the global registry, parents are entering data. In the CLIC registry, clinicians are entering their clinical data, and we have the opportunity to link those up if you allow us using what's called a CRID, and we can help you get one of those. But we're really looking forward to continuing to expand the clinical database. If you see one of the Click physicians and they ask you to enroll in the Click database, please consider that. We also expect this year to see some excellent progress on genetic therapies for PWS and SYS, bringing that closer to ultimately the clinic. And we're excited to be sharing Path for PWS data. Many of you have participated in the Path for PWS study. We have gotten all of that data in. We're busy analyzing that, and we'll be sharing that this year. So we're very excited about that. So all I have left to do, well, I have two things left to do then. I have to remind you, or we hope to see many of you in Phoenix. It's going to be a really exciting meeting. Scientific meeting looks fabulous. There's more than 150 abstracts that are going to be presented. The family meeting also looks great. So hope to see you there in Phoenix. And then the only other thing I have to do is thank you all again for your support, without which none of this would be possible. So we really appreciate everything you do for our PWS community, everything from filling out your surveys to running fundraisers. It's really critical. Thank you. So Melissa, with that, I will.
Yes. All right. Well, thank you so much for Teresa. You always make everything so understandable for our families, and myself included. So thank you. We have a bunch of questions. So let's get started. How does FPWR choose which grants to fund, and can parents play a role in how those decisions are made?
Yeah, sure. So we use both scientific experts and parent experts in our funding decisions. So we have a scientific review, and the scientists determine whether things are meritorious from a scientific standpoint, if they're innovative, if they're filling a scientific need. And then parents weigh in as well as advocate reviewers, and they tell us basically, is this important to me and others in the community? So it can be very good science. If it's not important to our families, we're not going to fund it. We're going to let somebody else fund that. But if it's both important to our families and scientifically sound, that's what we fund. So yes, we're always looking for advocate reviewers. Please, I think on our last slide, we had Jessica's email on there. She's probably the best person to email if you're interested in becoming an advocate reviewer.
Wonderful. Our next question is, can the cells developed in a hypothalamus in a dish be used to see how the hypothalamus may respond to drug treatments?
Yes, that's exactly the purpose. So it's not, it takes a little bit of doing to make a hypothalamus in the dish, and there's still some development that needs to go on. So we're funding the group at Harvard. We also have a team at Emory who are working on slightly different, but also looking at that question. So the ultimate goal of that is to get a reliable sort of hypothalamus in a dish, and then yes, test drugs on that, right? So right now, we don't have human brain that we can test things on except by putting them into your child. And we don't want to do that until we know that something is effective and it's safe. So this will allow us to really look at that efficacy piece anyway, and some safety issues in a brain in a dish. So it's a developing technology, and there's lots of advantages of using human cells in a dish as opposed to some of the other techniques that have been used for drug development before. So we're excited to be sort of in that in the pathway of making that happen.
Cool. The next question is, how many brain organoid projects have we funded so far, and how far along is the field of I can't speak, organoid development generally at this point, trying to get a sense of the practical utility in the near and medium term?
Yeah, I think I've sort of touched on that it is not sort of not anybody can, go and make a hypothalamus in a dish. There's still a lot of art that is involved. And I think, so as I mentioned, we're funding the group at Emory, we're funding a group at Harvard, all of whom are, and they're talking to each other, of course, as well. They're sharing reagents, they're sharing knowledge. Um, so I think we have a little ways to go before we can reliably, make that hypothalamus. The other thing is, any of these organoids, they tend to mimic uh, earlier in development. So there needs to be some work to make sure that what we're looking at as a hypothalamus in a dish is representative of kind of, what is in a person that walks around. So there's still some limitations to the technology, but it's come very far in the last few years, and I think it will continue to progress rapidly. So I think the concept of and the other thing is it's not easy to do, so you can't screen thousands of drugs on these organoids, but you can screen thousands of drugs on neurons in a dish. We're doing that. We know how to do that, and then take the best ones and really look at them in these organoids. So it's just a matter of prioritizing and getting a pipeline in place that makes sense with the available technologies.
Inspired by the cool images you showed in the neurovascular project, can we help by volunteering for brain imaging?
Well, that particular project, they're going to do in mouse brains, because the imaging that they do is better to do in a mouse, let's just say. So that particular imaging, no, but we're going to look forward to seeing what it shows in the mouse. Some of the other imaging, for example, the project at the University in Amsterdam, it is going to be done in humans. So, but that's in Amsterdam. So that's, I don't know where you are, but that may be a ways away. There are some other studies going on. For example, Laura Hulson is doing a study on transcranial direct current stimulation that includes an imaging component in PWS. She's done a lot of very nice work in imaging. There's a study in Cambridge by Stephanie Brown looking at using, individuals with PWS and looking at in live individuals, you can do certain scans that will allow you to look at some of the brain chemistry. So she's doing some studies looking at that and seeing how that relates to risk of mental health problems later in life. So there are a lot of really cool imaging studies ongoing in PWS. And I'd be happy if I think my email is on there. If anyone's interested in volunteering for any of those, there may be opportunities depending on where you're located.
The next two questions are about acetylcholine deficiency. Am I saying that right? So the first question is that can it be addressed with the use of currently approved drugs? Would a clinical trial in PWS be needed to test their efficacy and safety? And then the next person also asked regarding this, what is currently being used for, and are they being used in the US?
Sort of yes to all of those. So there are drugs that impact this system. I think the stepwise would be, first you need to show that the system really is important in PWS. So Dr. Yee has the images from the brains of PWS that shows that the system looks like there's a deficiency. She's going to move to showing that using brain imaging in people with PWS and correlating it with, what you would like to see when you think of this as a therapeutic is that, those individuals who have the lowest levels are also the ones who have the most challenges. And that would give you some confidence that that treating this may correct some of those problems. Yes, then you would need to look at safety and efficacy specifically in the PWS population. And to do that, you would have to, what are you going to measure? That's a big question. And that's actually an effort within FPWR. We are reviewing, so what cognitive tests make sense in PWS that people can do, that you can see change. There are many other neurodevelopmental disorders that are looking at different drugs to improve cognition. So we're taking our lead from some of those because what you would want to do is look at the safety. That's the most important thing. But then you would want a measure of whether these drugs actually do improve behavior, cognition, some of these things that we're interested in. So yes, those drugs are being used. They're being used sometimes in dementia, and they may be applicable to PWS, but we would take a very careful and stepwise series of experiments to get there. So don't go out and buy them today and give them to your adult with PWS, or certainly not your child with PWS. But it definitely could be a pretty quick or relatively quick transition to where they would be used in PWS.
Great. How do I know if my doctor is a PWS clinician?
So if you go on our website, and there is the PWS Clinical Investigation Collaborative, we have a page, and at the bottom of the page, it lists everybody, all of the physicians who are part of that collaborative. So I think there's about 25 to 27 sites in the United States. It's mostly doctors that are at, major academic centers, because those are the ones who are doing most of the research in PWS. But if your doctor is not a member there, and if they have an interest in joining that group, or being updated by that group, for example, you can contact me or Caroline at fpwr.org, and we will, we will contact them, we will talk to them, and put them in touch with the right people.
Great. The next question is, will PWS research be impacted by the federal cuts like those to the NIH?
The answer is very likely yes. So, you've heard today two of the investigators are at Harvard University. Well, we as a so $150,000 is a lot to us, but it's nothing compared to how much it costs to actually do this research. The technology that's used, the machinery, all of that already exists at those institutions because of the NIH. So the NIH, including their indirect costs, is what has built that infrastructure that we can leverage as a small organization. There's no way that FPWR can carry that research. So we are really dependent on there being a strong US biomedical infrastructure that we can use. That's been the model to date, right? You may have heard about the indirect costs. Well, we give 8% indirect costs, which is not nearly enough to cover the lights, the accountants, the oversight that our research projects require at a university. The reason that the universities let us do that is because they get indirect costs from the NIH. So the NIH has built this research infrastructure that we are then able to leverage. And if that NIH funding goes away, it's going to be disastrous for the ability of the scientists to get our work done, I believe. So I think advocating for a strong NIH, advocating for a strong FDA, the FDA has been cut. With thousands of jobs have been cut. We all know how long it takes to get FDA approval. I can't see where it's not going to start to take longer now that they've cut 20% of the workforce. So advocating for a strong NIH, advocating for a strong FDA. We don't, like, our research is not directly usually applicable to the National Science Foundation, but the imaging that our researchers use all came from grants through the National Science Foundation. So all of those things are really critical. I don't mean to go off on a tangent, but we require a strong biomedical infrastructure to get our work done here.
I appreciate you answering that for us. So among the ongoing clinical trials that we have, are there any that are nearing FDA approval?
Yeah, so the Acadia trial is a phase 3 trial, and so that trial is still ongoing, but I believe it's been enrolling well, and I believe they had a press release just recently, and I think they said, but you can go to their website and check. I think they said they expect results, in early 2026. So once they get those results, if they look encouraging, they will then submit their new drug application to the FDA. And hopefully the FDA will be able to review it relatively quickly. So I think that is the next one in line. FPWR is doing a study using a device, a vagus nerve stimulator. That approval process is different than for drugs. That device is currently not approved in the US, but potentially could be approved, either for PWS or for other things, in the next, well, I don't want to put a timeline on it, but in the short term, maybe a year, maybe 2 years, would be a 2 to 3 years would be a realistic thing there. And then that would be accessible to families. Aardvark is starting a phase 3 study. That'll take a little while to play out, but that's another one that's up and coming. So I think there are, and the pitolisant study, that is an already approved drug, and now they're looking at it in PWS. So that is another one. So there's many things in the cooker, all of which in the next year to 1 to 3 years, I think, what I would love to see is an entirely different landscape in 3 years where we have many different therapeutic options for our loved ones with PWS.
Well, thank you everyone for the wonderful questions. This ends our time. Teresa, we always appreciate you helping us digest this information, and thank you for all of your work. And thank all of you for joining us today. This webinar was recorded and will be made available shortly. Thank you so much.